The composite cell membrane is a multipurpose machine. In this lesson, we will build an intuitive picture of the plasma membrane, its cell adhesion molecules, and how the actin cortex couples to the membrane through talin, ezrin, and merlin. We will also see how lipid signals like PI45P2 and PI345P3 recruit proteins to the membrane, and briefly connect to receptor recycling. Start with the stratified structure. The plasma membrane is a lipid bilayer with a soft polymer brush on the outside called the glycocalyx, and underneath we find the actin cortex. We now add the bilayer. Next, we add the glycocalyx that keeps surfaces from sticking. Finally, the actin cortex provides mechanical support and helps drive shape changes and movement. Communication with other cells and with the extracellular matrix is handled by cell adhesion molecules, or CAMs. These include integrins, selectins, cadherins, and immunoglobulin superfamily members like ICAM and VCAM. A second set are large glycoproteins such as CD43, syndicans, and CD44. These act like Janus heads, as anti-adhesive brushes, and as adhesion mediators. Let's place a few representative CAMs into the membrane. Now add the glycoproteins with their sugar coats. CD44 can bind the giant polymer hyaluronic acid. This can hold cells apart over distances of around 100 nanometers. Watch a hyaluronic acid chain extending from CD44 to space the cell away from its neighbors. Beyond binding, the membrane itself generates repulsive forces through thermal undulations. These fluctuations create an entropic surface pressure that helps set intermembrane distances. Here, we gently undulate the bilayer to symbolize these thermal motions. How do CAMs couple inward to the actin cortex? Through proteins that expose Fe or M domains, including talin, ezrin, and merlin. Talin binds integrin beta chains and actin. Ezrin links glycoproteins to actin and even microtubules, while merlin can switch off actin coupling, acting as a tumor suppressor. Let's add these linkers under the membrane and connect them to their partners. In resting cells, these linkers often sleep in self-inhibited shapes. Activation exposes a polybasic stretch and a membrane binding pocket. Then electrostatic and hydrophobic interactions anchor them to PI45P2 or PI345P3 rich patches. We mark talin's sleeping state. A phosphorylation event flips talin to active, exposing plus charged segments. Negative lipid head groups recruit talin to the membrane. Talin binds the integrin beta tail, breaks a salt bridge, and switches integrin from low to high affinity. Membrane recruitment is often switched by local lipid chemistry. PI45P2 and PI345P3 densities are tuned by a kinase phosphatase tandem. PI3 kinase adds a phosphate to create PI345P3, increasing head group negative charge. We draw a small lipid control panel. When PI3 kinase turns on, negative charge rises, and proteins with plus rich patches snap to the membrane. One example is phospholipase C-gamma, recruited by a burst of PI345P3 anchors. Many receptors do cycles of endocytosis and recycling. The transferrin receptor is a classic example. After fusion with early endosomes, Rab5 and Rab4 guide rapid sorting and recycling back to the plasma membrane. We sketch a simplified path from plasma membrane into an early endosome and back out via a Rab-guided route. To summarize, the membrane integrates adhesion, signaling, and mechanics. CAMs like integrins, selectins, cadherins, ICAM, and VCAM connect outward to the environment and inward to actin via talin and ezrin, while merlin can turn off coupling. Glycoproteins such as CD43, syndican, and CD44 serve as anti-adhesive brushes and as receptors, even binding hyaluronic acid to set cell spacing. Lipid codes, especially PI45P2 and PI345P3, recruit sleeping proteins to the membrane by electrostatic hydrophobic forces, flipping adhesion and signaling on demand. Rab-guided routes recycle receptors efficiently. The composite cell envelope is truly a multipurpose machine.